Hello, my dearest students. How are you guys today? So, what we're going to be going over is the Blood Vessel Lecture 2, Part 2, which is going over the special circulation. So, if you remember from the previous blood vessel lecture, you had this as your handout. And you should have your handout printed and available to you so you can kind of track along with the lecture and answer some of the questions that we're going over during the lecture while you're actually watching the video. What we're going to be focusing on today are the special circulations. These are circulations that serve specific functions and often deviate from traditional circulation routes that you see in like the systemic circulation, so we apply the term special circulation to them. We're going to be going over the cerebral arterial circulation, hepatic portal circulation, and fetal circulation, and we'll get into that in just a minute. But if you want to track along with what we're talking about on the video, and you want to think about some of the things that are going to come up on the quiz over this particular material, I would have this handout out and available to you so you can answer the questions as we go along. It'll give you a guide to kind of frame what we're talking about. The other thing I'd have out and available while we're going through this is looking at your blood vessel lab. Any blood vessel, unless I expressly state this is going to be on the exam, that's not on the blood vessel lab isn't going to be on the exam. There's a few that I'm going to talk about with fetal circulation that are not on the blood vessel list. So just pay attention to that, but for the most part, if you memorize the blood vessels on the blood vessel list and you know the models and you watch the previous video, you have a pretty good idea of what to expect on something like a lab practical. Remember that the lab practicals are essentially purely anatomy based. It's image recognition and being able to say, hey, this is where that is, that's what that is. So without further ado, let's go ahead and get started with what our primary focal point is today, and that is on the special circulations. So the special circulations, number one that we're going over is the cerebral arterial circulation, also known as the circle of Willis. Your brain is a very metabolically or energetically needy organ. About 25% of your metabolic resources or your energy at any given point in time is used to fuel neurological processes. And as a consequence, the brain needs a lot of blood. It needs oxygen, it needs glucose, it needs those things to burn to make ATP. You need to flush metabolic waste out. So it's really important to get blood to the brain. It's also really important to regulate the blood pressure in the brain. And that's kind of what the circle of Willis does, or the cerebral arterial circulation. The other circulation we're going to be talking about is the hepatic portal circulation. Hepatic portal circulation is essentially a connection between blood draining from the GI tract, and notably places like the small intestines, and all that nutrient-rich, toxin-rich blood is first shunted or directed to the liver, and the liver filters and processes that blood before it's allowed into your systemic circulation. It's a very important mechanism to protect your body from just flooding it with things from the external environment, which is where food is ultimately coming from. It's also a processing mechanism that allows you to extract important things from the blood and allows the liver to do what it does. The final circulation we're going to be going over is fetal circulation. And when you think about the point of maternal and fetal exchange, so during gestation you have a developing embryo first 8 weeks, fetus for the next up until week 37 to 39. The point of exchange between the maternal circulation routes and the fetal circulation routes is the placenta. So we're going to focus on the placenta and that as a point of exchange, not mixture, but exchange. And then we're going to look at the blood flow networks within the fetus itself, and we're going to look at some of the differences between fetal and adult blood flow, because there are anatomical adaptations in fetal circulation that make that system more efficient. So, without further ado, let's start focusing about blood getting to the brain. Brain, again, hugely energetic organ. It needs a lot of energy and therefore it needs a lot of blood. So we need reliable routes in which to get blood to the brain. Now, the two major arteries that you can see externally on like the models that we're going to use in class, those skull models from this view, we have our right common carotid artery, and that branches to form the external carotid, which branches again to form a bunch of arteries that feed the external portion of the face and like the salivary glands. and the skull. And then we have the internal carotid, which feeds into what's called the cerebral arterial circulation. So that's bringing blood, or one of the major vessels that brings blood up into the brain. It also branches to form the ophthalmic artery, which takes blood to the eye, which is really important because the eye needs blood, notably the retina. Another really important artery in this particular situation is the vertebral artery. The vertebral artery branches off the subclavian artery, and when you think about the transverse foramen of the cervical vertebrae, what's running through those are the vertebral arteries and the vertebral veins, meaning that these are so important with respect to cerebral circulation that the body has dedicated metabolic resources to actually physically building bone structures around these arteries. So these are really important in supplying the brain with blood, and that's how they look externally. Now, we're going to focus on the circle of Willis internally, and in order to understand the circle of Willis and why it's so important, you need to have an understanding of this concept of an anastomosis. And an anastomosis is either a natural or a surgical joining of tubular structures, arteries, veins, your GI tract, those are tubular structures, so that they can become continuous. And here we're looking at kind of surgical anastomoses, but an anastomosis is essentially a connection, and the beauty of anastomoses in circulatory systems is if one blood flow network becomes occluded or blocked, there are alternate routes in which to get blood to that same region of the body. So it's just a connection between tubular structures, but the beauty of it in the cerebral arterial circle is that it allows for alternate blood flow routes if one blood flow network becomes occluded. And I'll kind of highlight what that really means. So a surgical anastomosis, an example I'll give you is, let's say that you had a blockage in the coronary arteries, and I don't know if you've ever heard of something called a coronary bypass surgery. But in coronary bypass, let's say you have a blockage in the anterior interventricular artery and in the right coronary artery. What you can do is you can actually remove a vein from a different part of the body, right? You can remove a vessel from a different part of the body, and you can essentially sew those on and create an anastomosis, meaning a network, and join the two tubes and essentially place this tube a little bit downstream from where the occlusion is in order to create an alternate blood flow network that can continue to deliver blood to these areas even though that coronary artery is occluded. So you bypass essentially that blockage in the coronary artery or in whatever artery you're talking about, and that's why this is known as coronary bypass surgery. In this case, because there are 2 points in which we're bypassing a blockage in the right coronary artery and the anterior interventricular artery, this would be known as a double bypass surgery, and it's just a way to ensure that blood is still getting to those parts of the heart. So anastomoses can be beautiful things, whether they're surgical or whether they're natural. It just provides alternate blood flow networks, and it's the joining of tubular structures so they can become continuous. And that's what a bypass surgery is. Now, when we look at the cerebral arterial circle, one of the best ways to visualize this is essentially wrapped around the sella turcica of the sphenoid bone. And we've done our bones and bone marking, so we kind of have an idea of where this is. And we're looking at this brain from the inferior portion kind of to the superior. So we're looking at the inferior portions of the brain where these blood vessel networks kind of connect to branch out to feed the different regions of the brain. Notably the cerebrum. Now, it's kind of hard to tell, but we're just going to kind of lay out the anatomy here. And then we're going to look at a different image. We're going to lay out the anatomy there, and we're going to figure out how this all ties together. So you have your internal carotid coming in through the carotid canal. You have your vertebral artery, so the internal carotid artery and the vertebral artery, which actually projects through the foramen magnum. Now, the vertebral artery feeds into this really important artery right here called the basilar artery. The basilar artery then branches out to form the posterior cerebral arteries, which will feed the posterior portions of the cerebrum, which is kind of like your higher order brain structure where you do a lot of your thinking, where your thinking happens. These communicating arteries that you see essentially connect blood vessel networks. They are anastomoses. Here we have the internal carotid. The internal carotid will eventually branch to form the middle cerebral arteries, which will feed different cerebral structures kind of in the middle portions like the temporal and the parietal lobes. Then you have the anterior cerebral artery. The anterior cerebral artery will continue up to feed the anterior portions of the cerebrum, such as your frontal lobe, which is where you have like where the processes of executive function and decision making and reasoning come from. These blood vessel networks are connected by what are called the communicating arteries. You have this very small little artery that connects the two anterior cerebral arteries called the anterior communicating artery. And then you have this one back here which connects the posterior cerebral to the middle and internal carotid called the posterior communicating arteries. Now, the beauty of this system is if one of these arteries becomes plugged up, there are alternate blood flow routes that can get blood to those same regions of the brain, and that is an important thing, right? That is the beauty of the circle of Willis. If one blood flow route becomes occluded or blocked up, or there's like a blood clot that forms or a plaque that blocks off blood flow, there are alternate routes of blood flow so you can continue to get blood to those areas of the brain and prevent neurons from dying, essentially, from brain tissue from being destroyed. So that's really important. The other thing that this kind of network does, this cerebral arterial circle does, is it dissipates pressure and it helps to regulate blood pressure in the blood vessel networks of the brain, because you don't want those high pressure systems potentially rupturing blood vessels and causing brain aneurysms. So there's a double whammy. Provides alternate routes of blood flow. That's function number one that I want you to know of the cerebral arterial circle, or the circle of Willis. And two, it helps to dissipate pressure and regulate blood pressure in the blood vessel networks of the brain. When we look at the model that's going to be used in class, this is the model, and this is essentially our cerebral arterial circulation model, or our circle of Willis model. You can call it either one on the exam. So what you have here, coming in through the foramen magnum, essentially projecting from those transverse foramen and the cervical vertebrae, these are the vertebral arteries. The vertebral arteries then fuse to join the basilar artery. The basilar artery then branches off, and here you have what are called your posterior communicating arteries. So these little ones are your posterior communicating arteries. This big one projecting kind of toward the posterior portions of the brain here is what's called your posterior cerebral artery. So vertebral arteries to basilar artery to the posterior cerebral arteries. We also have our cerebellar artery right here, which feeds a really important brain structure called the cerebellum, and other branches of the basilar which feed brainstem regions, but they're not on your list and I'm not worried about them, although they're important. So you have your posterior cerebral arteries, your posterior communicating arteries, which connect essentially the blood flowing from the basilar artery to the blood flowing through the internal carotids. Now, the internal carotid arteries eventually branch out to form the middle cerebral arteries. They will also form, you see these little guys projecting through the optic foramen? Those are the ophthalmic arteries, and the ophthalmic arteries feed the retina of the eye. So you need those in order to provide nutrient and oxygen-rich blood and to rinse out the metabolic waste from the retina of the eye, which allows you to see. And you can actually see the ophthalmic arteries by dilating the pupil and looking at the retina with an ophthalmoscope. You can actually see the pulsations. And those are your optic nerves right there running through the optic canal. So that's a really important feature. These are your anterior cerebral arteries, which are feeding the regions of the frontal lobe, which are incredibly important for like personality and executive function. So you can actually kind of predict what you would expect to see in the change of a person's demeanor or behavior if they had something like a brain aneurysm or a stroke based on the regions of the brain that are affected, which you can tell by where the blood vessel networks have been affected. This little artery that connects the two anterior cerebral arteries together is called the anterior communicating artery, and it's a really important one to know medically because it's at the point where a lot of problems can arise, as you're going to see in just a minute. Now, those are the carotid canals. Running through so there's the foramen rotundum, the foramen ovale, and this would be the foramen spinosum. What's running through the foramen spinosum are actually what are called the middle meningeal arteries, and they're not feeding portions of the brain, they're feeding the connective tissue networks that support the brain and spinal cord. So, circle of Willis. If one of these becomes clogged up, let's say that you had a blood clot or a occlusion of the vertebral artery, this other vertebral artery would be able to feed into this circle of Willis, and you would still be able to get blood flow, although be it reduced blood flow, to the same regions of the brain. That's really important. Two, this circular formation and the fact that these blood vessels kind of branch out and dissipate out helps to lower pressure and regulate the blood pressure of the blood flowing in the cerebral arterial circulation, again, which is very, very important. The model you'll get on a practical is this one. We're talking top down, baby. So I want you to pay attention to this video to answer a few of the questions that I've asked you. Oh, and it went off. There we go.
This is Dr. Cal Shipley with a review of the circle of Willis, Berry aneurysms, and subarachnoid hemorrhage in the brain. To begin, let's take a look at the anatomy of the circle of Willis. The circle of Willis is best seen by examining the underside of the brain. The circle of Willis is really a beautiful piece of anatomy and represents a central hub from which radiates the entire blood flow to the brain. Inflow is primarily via the internal carotids and vertebral arteries. Outflow of blood from the circle occurs via anterior, middle, and posterior cerebral arteries, each with left and right branches on either side of the circle. The basilar, posterior communicating, and anterior communicating arteries complete the circle. By merging the inflow of blood to the brain from the carotid and vertebral arteries, the chance is minimized that an obstruction or impairment in flow in any one of these arteries will result in a significant impairment of blood supply to the outflowing arteries. Now that we have a better understanding of the anatomy of the circle of Willis, let's take a look at how it contributes to subarachnoid hemorrhage in the brain. While beautiful in design and function, the circle of Willis is also the location of 85% of all aneurysms within the brain. One common site for the occurrence of an aneurysm is the junction between the anterior communicating and anterior cerebral arteries. Aneurysms form as a result of an acquired structural weakness in the blood vessel wall. Arteries which carry very high flows, such as those in the circle of Willis, are thought to be particularly susceptible to mechanical damage in the vessel wall. Contributory factors such as cigarette smoking or high blood pressure may accelerate the process of damage to the vessel wall. The strength of the aneurysm wall is inversely proportional to its size. Lesions 1 centimeter or larger are particularly susceptible to sudden increase in growth and rupture. With rupture, blood spreads rapidly across the surface of the brain via the cerebral spinal fluid. Bleeding generally only lasts a few seconds. However, rebleeding may occur and is most likely within 24 hours of rupture and initial hemorrhage. The most common cause of death and disability after subarachnoid hemorrhage is vasospasm, extreme narrowing of the arteries with impairment of blood flow to brain tissue, and subsequent stroke. Vasospasm usually occurs within the first 3 days after hemorrhage and generally peaks in effect about 1 week out. Vasospasm occurs as a result of chemical irritation of blood vessel walls by breakdown products of hemorrhaged blood. The duration of vasospasm and the location of the vessels affected determines the size and depth of subsequent strokes.
So what they were really saying in this video, and we'll go back to the circle of Willis, is why do we care about high blood pressure? Because when blood pressure is high, it puts stress on blood vessels. And I want you to think about blood vessels being kind of like a water balloon, right? A water balloon is designed to withstand a certain amount of pressure, but if you put too much pressure on that water balloon by filling it up too much, it's going to burst. And the same thing happens particularly in small blood vessels. So when you explain to a patient why high blood pressure matters, it's because it's ripping apart their blood vessels internally, and it's putting them at an increased risk of stroke and increased risk of aneurysm. When we have this aneurysm, which is where 85% of subarachnoid hemorrhages occurs at the anterior communicating artery, because it's just a little guy, but it's subject to a lot of pressure fluctuations. All of that material that's released, even though the bleed only lasts for a little bit, it causes vasospasm or vasoconstriction in these blood vessels. Can you imagine why a damaged blood vessel, the chemicals may cause vasoconstriction or vasospasm, thinking back to our hemostatic pathways? Well, when you damage a blood vessel, it releases chemicals that trigger vasospasm. Vascular spasm is an adaptation to a damaged blood vessel. That's a damaged blood vessel, and all that stuff is leaking to other blood vessels and causing them to go whoosh and clamp shut. When they all start clamping shut, that increases the chance that clotting mechanisms are going to activate, and you're going to have strokes all over the place. With a stroke, which is a blood clot, you're not going to get blood to certain regions of the brain. Those regions of the brain are going to start to die, and that's going to be massively problematic. Now, with any system of arteries, you have to bring blood to the brain, but you also have to get blood back out of the brain. When you think about that, there are common drainage points of blood that's fed to the structures of the brain. A couple of common drainage points that are going to be important are the superior sagittal sinus. A sinus, when you're talking about the cardiovascular system, is just a big fat daddy vein. And then we have this transverse sinus, and I think those are the only two that you need to know. And the common drainage point for the superior sagittal and transverse sinus and all of these sinuses, but there's only two that are on your list that you need to know, is the internal jugular vein, and then that jugular vein projects out the jugular foramen of the temporal bone and ultimately fuses back with the subclavian, which becomes the brachiocephalic, which becomes the superior vena cava, and that blood goes back to the right atrium of the heart. Two that you need to know, superior sagittal sinus and transverse sinus. They are incredibly important common points of drainage for the cerebral circulation. So that's the cerebral arterial circle. Now we're going to jump into what a portal system is. So we're going to talk about hepatic portal circulation. And in order to understand hepatic portal circulation, you first need to understand what a portal system is and how a portal system deviates from typical blood flow. So in a typical system of blood flow, arteries eventually branch into smaller blood vessels called arterioles. Arterioles feed into capillaries, which are points of exchange between the tissue. Capillaries are drained by venules. Venules fuse to form veins. Veins bring blood back to the heart. And that's a typical system. That's a typical blood flow route in your systemic circulation. Remember that when you're tracing out a drop of blood. Give me the capillaries, because I want you to know that that's the turnaround point and the point at which the arterial system interacts with the venous system. In a portal system, arteries branch into arterioles. Arterioles feed into capillaries. There's exchange at a tissue. Those capillaries then drain into another vessel. It could be a vein, it could be an arterial, but they drain into another vessel where they are taken to another set of capillaries in a different part of the body. Why would you want capillaries in one part of the body connected to capillaries in other parts of the body? So whatever's happening here, you can chemically link the events in one part of the body to the events in another part of the body through these exchange networks, meaning that these are points of exchange, essentially. It's a way to take materials from one part of the body to another part of the body directly without having to go through the entire systemic circulation and get filtered out by things like the kidneys and the liver. It's a connection point. Those capillaries are then drained by venules, which go to veins and then back to the heart. So when one capillary network is connected to another capillary network by whatever vessel is connecting them, that's called a portal system. Essentially, it's a port right, between one network of capillaries and another network of capillaries. When you think about hepatic portal circulation, this is hepatic portal circulation in a nutshell. You have the blood feeding into the GI tract, such as the stomach and the intestines. And along the GI tract, and I'll focus on the small intestines here, we have nutrients absorbed. So you eat something or you drink something, and things like glucose and amino acids and vitamins and minerals and water are being absorbed, and sometimes toxins and bacterial pathogens and whatever else is being absorbed because we're taking this stuff in from the external environment. And it's being absorbed into the blood vessel networks that line the GI tract, and specifically into those capillaries. Now, those capillaries, so we have artery to arterial, arterial to capillary, those capillaries are going to be taken by a vein called the hepatic portal vein, because it's part of a portal system, to another network of capillaries within the liver. So all the blood that leaves the GI tract is first shunted to the liver, and in the liver, that blood at those sinusoidal capillaries interacts with liver cells, and within the liver, in these sinusoidal networks, three things happen. That blood is filtered for toxins leaving the GI tract, so we filter that blood for nasty stuff we don't want spreading into our systemic circulation. It's called first pass metabolism. The second thing is that the hepatocytes are going to process nutrients, so they're going to take in glucose to make glycogen. They're going to take in amino acids to make things like proteins. And what that allows the liver to do is not only process that blood, but it allows the liver to introduce things like albumins and clotting factors into the bloodstream. All of those capillary networks are then drained by the hepatic veins, not the hepatic portal vein, but the hepatic vein, which is then drained into the inferior vena cava, taken back to the right side of the heart where it enters pulmonary circulation, to the left side of the heart where it's going to be pumped in systemic circulation, and now we have filtered and processed blood. Long story short, we connect the blood vessel networks of the GI tract to the blood vessel networks of the liver so we can filter and process blood before it's allowed to enter into our systemic circulation. That's it. We're connecting the blood vessel networks of the GI tract with the blood vessel networks of the liver in order to filter things out. So when you think about this and you tie these diagrams together, this would be the lumen of the intestines, meaning you eat something, it starts its digestive processes in the mouth, and its mechanical and chemical breakdown into constituent particles, things like glucose and amino acids and nucleotides and whatever else. Those are absorbed along the lining through the cells of the GI tract, and they're absorbed into the bloodstream, the capillaries within the GI tract. When you look at the capillaries within the GI tract, you have, right, these capillary networks going from being red to blue, so they're supplying these cells with oxygen and nutrients that they need. They're also absorbing or taking all that stuff that's absorbed along the GI tract, and then it enters into venules. These venules will ultimately fuse to form veins, and those veins will ultimately all come together to form the hepatic portal vein, which is going to take that blood to the liver. It's kind of tying this to the systematic. We look at the cellular and the molecular level, then we look at the systems level, and we go, okay, so all the blood leaving the GI tract and the spleen first goes to the liver, where it can be filtered and processed and have proteins introduced to the blood before it's allowed to enter into systemic circulation. The system that allows us to do this is called the hepatic portal system, and being able to label the blood vessels of hepatic portal circulation is really important. In fact, I ask you to do it on your diagram. So when you think about your lab, a lot of these blood vessels are actually asked within the context of your lab. Now, I always think of hepatic portal circulation as looking kind of like a chair. The back of the chair is the hepatic portal vein. Notice that the hepatic portal vein is purple. It's not blue or red. It's indicating oxygen intermediate blood, and I like to think of it as being nutrient-rich blood as well. So we have the hepatic portal vein. The back of the chair is the superior mesenteric. The front of the chair is the inferior mesenteric, and the seat of the chair is the splenic vein. And long story short, you're not going to have to know specific regions, but the superior mesenteric and inferior mesenteric take blood from the GI tract, drain that nutrient rich, toxin infused blood from the GI tract, and they bring it, right, up to this hepatic portal vein. All the blood leaving the spleen is also directed via the splenic vein to the hepatic portal vein, and then the hepatic portal vein takes that into the network of sinusoidal capillaries within the liver itself. So you have capillaries here, you have capillaries here. That's why it's called the portal network. When that blood gets into the liver, it percolates through the liver. Hepatocytes remove things like glucose and amino acids, so they process that blood, right? They introduce proteins. They also filter that blood for nasty things like toxins and potential pathogens that may have entered into the GI tract. And there's a special type of white blood cell within the liver that even targets pathogens. It engulfs them and gobbles them up and destroys them. Once that filtered and processed blood has percolated through the liver, which you can kind of think of like a blood filter, right? It enters into the hepatic veins. Do not confuse the hepatic portal vein, which is connecting one capillary network to another capillary network, with the hepatic veins. The hepatic veins are traditional veins that take all that filtered and processed blood to the inferior vena cava, which then goes to the right atrium to undergo pulmonary, and then left part of the side of the heart to undergo systemic circulation, where that filtered and processed blood is allowed to move through the body. On lab models, you can see that chair structure pretty readily. So this is the posterior aspect of the pancreas right here. Here's the spleen. And when you look at it, the back of the chair is the hepatic portal vein that would be going up to the liver. What you would consider the back of the chair. The back leg of the chair is the superior mesenteric. The front leg of the chair is the inferior mesenteric, which are common drainage points of the capillaries and veins that are leaving or draining that nutrient-rich blood from the GI tract to be filtered and processed by the liver. And then you have the splenic vein, which is bringing all that blood from the spleen to join with this hepatic portal circulation. That hepatic portal vein will then enter into the liver. It will enter into a series of sinusoidal capillaries that allow for the filtering and processing of blood. And eventually, once it goes through the liver, this is a top-down view, that blood will drain into the hepatic veins. The hepatic veins then directly drain into the inferior vena cava, which, by the way, essentially runs right through the liver. You have a huge amount of blood flowing through the liver, and if there are any blockages in blood flow or bile flow in the liver will actually swell, and you can feel that. So now we get into fetal circulation, and I want to put fetal circulation into context for you. So a sperm and an ovum, or an egg, fuse, right? They form the first cell, the first diploid cell, meaning the first cell with 46 chromosomes that ultimately has the potential to become a life, called a zygote. That zygote starts to divide like crazy, forming this mass of cells that's eventually implanted into the uterus. Now there's different layers in this mass of cells, and one of these layers ultimately goes on to form the placenta. The placenta is the point between maternal and fetal exchange. It's also the structure that releases a hormone called human chorionic gonadotropin, which we use as a marker of pregnancy. From week 0 to week 8, that developing life is referred to as an embryo. After week 8, all of the major organ systems have been formed, and now that particular life from week 8 to week 37 to 39 is referred to as a fetus. So what we are looking at here is fetal circulation, and the point of exchange between the maternal circulation and the fetal circulation takes place at this incredibly fascinating blood vessel-rich structure known as the placenta. There is something I absolutely want to highlight here. Maternal and fetal blood don't mix. Maternal and fetal blood don't mix. There is an exchange between maternal and fetal blood, but they do not mix. If they mixed, if you had a mother who was type O negative and a gestating baby that was type A, if the blood mixed, the antibodies from the mother could attack the antigens on the surface of the baby's red blood cell, and it could cause a massive problem and a spontaneous miscarriage. All sorts of other problems. Maternal and fetal blood don't mix. There is exchange between maternal and fetal blood, right? So things like oxygen and nutrients are delivered into the fetal circulation, and the fetus exchanges back with the maternal circulation. It's how it gets rid of waste and carbon dioxide. It's also how it sends chemical signaling cues. It says, hey mom, I'm hungry. Hey, I need this or I need that, in the form of chemical messengers. So there's an exchange back and forth. Right? It's not just a one-sided exchange, and that's kind of the interesting thing. That exchange is what helps guide fetal development, by the way. So the point of exchange between maternal and fetal circulation is at the point of the placenta, which is an incredibly fascinating organ in and of itself. It is one of the most amazing things that I've ever studied. It's incredible. Now, fetal circulation is different than the circulation of a newborn, which is different than the circulation of an adult. Adults and newborns have to live independently. They have to get their nutrition and their oxygen and their nutrients on their own. The fetus is dependent on the maternal circulation for things like oxygen and nutrients, and therefore it needs to have adaptations that essentially allow for maternal-fetal exchange and also shunt blood away from non-functional organs. And in fact, I would say the major function of the fetal circulation is to shunt or direct blood away from organs that are not yet functioning, because it would be a waste of resources to deliver blood to organs that are not functioning. So it shunts blood, or it directs blood. When I say shunts, I mean it directs or causes blood to move away from organs that are not yet functional. Now, when we trace out fetal circulation, what I want you to imagine here is a baby. So around this diagram, you got a little fetus developing, right? You got a potential life in there that's developing. And the point of exchange, so this fetus is still within the amniotic sac. It's still gestating. And the point of exchange between the fetus and the maternal circulations is at the placenta, and that's where I'm going to begin. It's also where I'm going to end. So we get exchange, not mixing, of maternal and fetal blood, never mixing, but exchange between maternal and fetal blood, and oxygen and nutrients are delivered into this blood vessel you see right here, indicated by number 1, called the umbilical vein. It's called a vein because it's bringing blood back to the heart, so don't let that red coloration fool you. This is the umbilical vein. The umbilical vein then delivers blood to the ductus venosus. The ductus venosus bypasses the liver and essentially takes that blood directly to the inferior vena cava, and it mixes the oxygen-poor blood coming out of the hepatic veins with the oxygen-rich blood from the umbilical vein to form an oxygen-intermediate blood that's then returned to the right atrium. We also get the superior vena cava and the coronary sinus bringing deoxygenated blood to the right atrium. From the right atrium, because pressure in the right atrium is higher than in the left atrium as a consequence of this circulatory route, there's a special adaptation that's indicated by number 5, and it is a hole in the atrial septum, not the interventricular septum, but the atrial septum, called the foramen ovale, or the oval hole. Why would you want a hole between the right atrium and the left atrium? Well, the right side of the heart is pumping pulmonary circulation, but a developing fetus isn't inhaling and exhaling, and therefore there's no exchange happening in the lungs. So why would you want to deliver a bunch of blood to the lungs? It's just going to decrease the efficiency of the system. So you want that oxygen intermediate blood going directly from the right atrium to the left atrium, where it can enter into systemic circulation, right? So you want it going directly where it can enter into systemic circulation. Any of that blood that ultimately gets into the right ventricle and is shunted into the pulmonary trunk and pulmonary arteries is actually, there's another adaptation called the ductus arteriosus, which is essentially an anastomosis that connects this to the aortic arch, connects the pulmonary circulation to the systemic circulation. So there are two mechanisms to prevent that blood from going to the lungs, because the lungs are non-functional organs at this point. So now we have the umbilical artery, which is oxygen poor or oxygen intermediate, delivering oxygen to the different tissues. And that umbilical artery eventually comes and it loops around the umbilical veins, and it'll eventually come back to the placenta. When it comes back to the placenta, what the umbilical artery does is it gets rid of carbon dioxide and metabolic waste. It also delivers any chemical signaling molecules that can diffuse across that placenta that say, hey mom, eat some chicken. And then we get the exchange again of the oxygen from the maternal and nutrients from the maternal bloodstream coming in, and that's fetal circulation. So go over fetal circulation a few times, label those. Now, once a baby is born, a few interesting things actually happen here. So once a baby is born, there are a series of chemical signaling molecules called prostaglandins that create a change in which the different structures become different things. So the umbilical vein and the umbilical artery become the round and the umbilical ligaments. So the round ligament stabilizes the liver, the umbilical ligament does some other things in the body. The ductus venosus becomes another ligament. The foramen ovale becomes a structure called the fossa ovalis, meaning that shortly after birth, that hole heals over. If that hole doesn't heal over, you have what's called a septal defect. And that septal defect actually has to physically be repaired by somebody like a cardiologist because it decreases the efficiency of the system. And we talked about that whole thing with the tetralogy of Fallot and the first surgeon that ever corrected it, the African-American surgeon, Vivian Thomas. So that heals over and it becomes the fossa ovalis. So all of these things that were in the fetal circulation eventually become ligaments, or they heal over, and that's what produces our adult circulatory system. If they don't, for example, septal defects are when the foramen ovale fails to close up. Patent ductus arteriosus is when the ductus arteriosus fails to heal up, and that can cause high pulmonary blood pressure, and it can cause other problems. And one of the things that is necessary for this to happen is prostaglandin. So there are medications you can give to actually cause this to heal over, or you can go in and you can surgically intervene. Long story short, know your fetal circulation and know the purpose of it. It shunts blood away from non-functional organs to make the circulation within the fetus a bit more efficient and to allow for maternal and fetal exchange. Not mixing the blood, but exchange. Thank you. Thank you so much. Thanks.